the JAMA Network.
Hello, my name is Bridget Fernandez. I'm a medical geneticist working in St. John's, Newfoundland, and I'm a professor of genetics and medicine at Memorial University of Newfoundland. The autism spectrum disorders are remarkably heterogeneous in terms of their clinical presentation as well as the underlying genetics. And we estimate that there are over 100 different types of genetic and genomic variants that can predispose a person to the development of an autism spectrum disorder. And while we don't know all of these, there's been tremendous progress made in identifying them over the last 5 years because of the introduction of high throughput forms of sequencing. Our objective was to look at the diagnostic yield of 2 relatively new forms of genome-wide testing in a sample of children with autism spectrum disorder. The study sample consisted of 258 unrelated children with autism spectrum disorder from Newfoundland. We recruited them consecutively from the 2 developmental pediatric clinics here that perform multidisciplinary assessments for ASD. Each child had a very careful physical examination looking for minor physical anomalies. These are slight morphologic differences that occur in about less than 5% of the population. Many of us have 1, 2, or possibly even 3 of these, but when there's more than that number, we start to wonder whether there could be a genetic difference in that person that expressed itself when the person was developing in utero. We classified the children into 3 groups of increasing morphologic severity. The 3 groups were essential, equivocal, and complex. We found that about 20% of our sample fell into the complex group. When both tests were applied, the microarray and the whole-exome sequencing, we found that 16% of the children had an ASD-related molecular finding, and that the chance of a positive genetic test was highest in the morphologically most complex group, where the diagnostic yield was over 35%. We were really encouraged by the results in the sense that, okay, we found 16% of the children with a positive genetic test, and really what we considered to be a very high number in that complex group. Going forward, our hope is to that eventually we and others will get to the stage where most of the genetic and genomic variation that underlies susceptibility to ASD will be known. And this will allow us to potentially then use genetic testing to make earlier diagnosis